envirominstitute.org, or Facebook, where we are now. And also you can find us on Twitter, and you can use the hashtag #ThisIsEnvirom. Also, we want to make sure you are aware of the opportunity to participate in the new study, the ECO study. It stands for Environmental Exposures and Chronic Health Outcomes. In this study, we're looking at the relationships between environmental exposures and chronic health outcomes, like heart disease, diabetes, liver disease, etc., among people who live here in the Louisville, Kentucky metropolitan area. And there are 4 total study visits and 1 phone call. Participants will be compensated $50 for each study visit and $25 for the phone call. So that's $225. If you are interested in learning more about participating in this study, please contact the study coordinator, Carrie Standiford, at 502-852-7559. Or you could email at ctu@louisville.edu. So the last time we were together, Dr. Lincoln Larson shared about his research on perceptions of greening in the Green Heart study. This week, we have a couple more exciting developments to discuss. We have 3 special guests today: Rebecca Turney, Dheeraj Kanaganti, and Ibrahim Juja. These are standout students who interned with us this summer, and they will share about their experiences here at the Envirom Institute. After the interns, we will receive an update from Lauren on COVID and the Omicron variant in Louisville. But before we get to all that, let me allow our communications team to introduce themselves. I am joined today by my colleagues and fellow Envirom Institute researchers, Dr. Natasha Desjarnette, Dr. Jason Hellman, Lauren Anderson, and Lainey Taylor. If each of you could introduce yourselves. Natasha?
Sure. Thanks for the kind introduction, Alex. I'm Natasha Desjarnette, assistant professor here in the Envirom Institute.
Hello, my name is Jason Hellman. I'm an associate professor of medicine in the Diabetes Obesity Center here in the Envirom Institute. My lab is interested in understanding biological mechanisms by which inflammatory tone and duration are modified in cardiometabolic diseases.
Hi everyone, my name is Lauren Anderson. I'm a research manager for the Center for Healthy Air, Water, and Soil at the Envirom Institute. We study how the fundamental building blocks of our world, air, water, and soil, impact health and well-being here in Louisville.
Hi everyone, I'm Lainey. I serve as a civic research aide in the Center for Healthy Air, Water, and Soil, and really am interested in how we can connect the community in ways that we're doing like today to the work of the environment, how we can all collaborate together on this.
And I don't think I mentioned, I'm Alex Carl, but I'm an assistant professor here in physiology. And I research the cardiovascular effects of inhaled toxins, particularly e-cigarette aerosols and volatile organic compounds, looking at cardiac pathophysiology in relation to those exposures. So with that, I think I'll hand the torch off to Dr. Hellman. Jason?
Thanks, Alex. Yeah, as you were mentioning earlier, we're really excited to have 3 outstanding interns that spent their summers with us. We have Rebecca Turney, who's an undergraduate student in the sustainability program, who's been interning with the Center for Healthy Air, Water, and Soil this summer. She has brought extensive GIS and mapping skills to the team and has helped to create an environmental vulnerability index for Louisville, as well as reports detailing the geographic spread of COVID-19. Thanks for joining us, Rebecca. We also have Dheeraj Kanaganti, who is a senior at Manual High School here in Louisville. He's been interning also in the Center for Healthy Air, Water, and Soil since last fall. Dheeraj has done some outstanding work, and he's built multiple machine learning algorithms to solve complex issues such as wastewater treatment flow and streamline the identification of open online data sets. Ibrahim Juja is joining us today from Emory University. He spent his summer in the Diabetes and Obesity Center, participating in the American Heart Association sponsored Biomedical Sciences Summer Internship Program. During this experience, Ibrahim worked alongside one of our faculty members investigating the biological actions of carnosine in striated muscle cell function. So thank you all very much for willing to join us and talking with us today.
Yeah, it's really great to have all of you with us so we can learn from you about what the summer held and really how it changed your view of health and really what you see yourself doing in the future. And I think to kind of continue with the introductions, we can start with Duraj on this, just to give everyone some understanding. It'd be great to learn where you go to school and what year you are currently in.
Awesome. Hi, everybody. My name is Duraj. I'm a senior at DuPont Manual High School, so right across the street from the U of L campus. And, I've been working at CHAWS for, as Dr. Hellman said, for about a year now. And it's been an incredibly enjoyable experience, very enriching, of course. And, just some things I do in my free time. I love to watch football. I enjoy creative writing, and I'm also big on hiking in the outdoors.
Awesome. Great to learn more about you in terms of where you're going to school and just you as a human. I see Rebecca next on my screen, and then we'll go to Ibrahim.
Yeah, my name's Rebecca. I'm a senior sustainability major and biology minor at U of L. I've been working with the CHAWS team since the summer, so I've had the opportunity to work on a couple projects, and I've really, really loved my time. I don't know, interesting things about me. I guess I have 3 cats. I play guitar, and I'm really, really passionate about sustainability. So.
Hello everyone, my name is Ibrahim. I also went to DuPont Manual High School, just like Dhiraj, and I graduated 2 years ago. And I'm currently a sophomore at Emory University. Some things about me, I really like the show Survivor. I run the Survivor Club here at Emory. We host like a mock Survivor almost. I also have a cat, just like Rebecca. And what else? I'm also an RA this year, so that's been a really fun experience. I just got done with training and I met my residents, and it's been really great so far. I love all of them.
So great to learn more about each of you. And I know this summer probably held so much for all of you, but if you could give us, just for everyone listening in today, we would love to learn more about your summer internship project. We know that there's probably a lot of different projects during the summer, but you had to give us like kind of a minute summary of what that held for each of you. How would you describe it to us? And we'll reverse the order and we'll start with Ibrahim this time.
All right, cool. So as Dr. Hellman mentioned, I think, I mainly worked on carnosine, which is a dipeptide. I worked in Dr. Baba's lab over the summer in the DOC, and I mainly looked at how carnosine would affect myosin in muscle tissue. So myosin can exist in 2 configurations, SRX and DRX. SRX is very slow, it's called super relaxed, and disordered relaxed is DRX, it's much faster. But both are slower when compared to active muscle fibers. But it's like 2 alternate states that it can exist in. And SRX and DRX is in a ratio in the heart. And studies have shown that this ratio kind of changes based on different conditions. Like for example, there's high DRX and hypercardiomyopathy and HCM, and it's also high DRX in failing hearts. And on the other hand, there's high SRX in the hearts of squirrels during hibernation. So this ratio kind of changes in the heart based on the conditions. And I wanted to look at how carnosine would affect the SRX-DRX ratio in the heart. Do you want me to go over like the results of my study as well, or just kind of like an overview of my background?
Yeah, that's a great question. If you want to share, maybe I think the next question can kind of lend itself to you talking about the results. So maybe we'll wait till the next question. But I think that's a really good summary of what you were working on this summer.
Can I ask a clarifying question, Ibrahim? Carnosine. So, I mean, you said it's a dipeptide, but like where do you find it? What are sort of the real world, I don't know, occasions where you would encounter carnosine?
Yeah, so carnosine synthase exists in, I believe, like all mammals and most living creatures, and it synthesizes beta-alanine and histidine to make carnosine. So in order to increase carnosine, like in animals, we like feed mice beta-alanine, and this high beta-alanine concentration in the mice causes a high carnosine concentration. but it's mainly found in, I think, a muscle tissue. That's the main place where it's found, and it's like naturally occurring in most all mammals and most living creatures, I believe.
So like ingestion of carnosine in, just everyday eating may have some health impacts, health benefits. Is
Yeah.
that sort of the presumption at the outset of your studies, or?
Well, I wasn't involved in this, but Dr. Baba did do some trials where he had people take carnosine pills or had like increased their beta alanine like supplements to kind of increase carnosine. But the results are, still being processed. I don't want to like say anything for sure, but
Yeah.
The goal, I guess, is to kind of see how ingesting carnosine can help people like in the future. That's like the long-term goal, sort of.
Yeah, I mean, I want to say, I think that carnosine is found in high levels in like chicken and certain meats. So it may be
it's in,
that,
like muscles.
right. So people who consume meat may get higher levels of carnosine, or I guess it's carnosine intake, as opposed to the beta alanine, which is a precursor to carnosine synthesis internally. But all of this, may relate to, everyday behavior of humans in terms of what they're eating. I take it.
So yeah, we have been looking at like how increased levels of carnosine, like people have increased levels, or I guess like right now it's mice, but mice have increased levels. How does that affect like their heart mainly is what we're focused on. How it affects like those mice with heart failure, mice without heart failure, like their lifespan, all that kind of stuff.
Cool. Thanks. That's all I got for now.
Yeah, as someone who, in full disclosure, Googled how to say carnosine before this call, like it's very nice to learn about all of this. Rebecca, we'll turn it over to you now to talk more about your summer.
Yeah, so the first project that I worked on was the noise pollution project, where I wrote a research memo on the correlation between noise pollution and PTSD for Congresswoman Donna Purvis. I also worked on the Scholars in Residence project, just drafting a website for the program and business cards. Definitely the biggest project this summer was the Environmental Vulnerability Index. Basically, kind of taking a study from Houston and using it as a template and applying it to Louisville to evaluate different areas of vulnerability and how different census tracts across Louisville are affected by it. My work on that was pretty GIS heavy. So GIS is Geographic Information Systems. You can take data basically and show the spatial relationship. It's a mapping program. So you take the data and you can display it on maps and show the relationships between different sets of data. So using that, we could physically see which census tracts are more at risk in Louisville. And then more recently, I've been working on the health geography report, tracking the COVID levels in the wastewater across the city, and trying to figure out maybe why, or just taking into account the different levels across the city. So that's what I've been doing this summer.
Sounds like a super not busy summer for you.
Yeah, very planned.
Lainey, do you mind if I interrupt with a question?
Yeah, go for it.
Rebecca, this is very interesting. So when we look on a map of Louisville, what vulnerabilities stand out?
Yeah, so Louisville is a pretty historically segregated city because of redlining, and those areas just hold up today, even though the policies are still out of place. So obviously there's like the 9th Street divide between East and West Louisville. The East End has more communities of like white, rich people mostly, and the West End has more communities of color and low income communities. There's also Rubber Town kind of on the left over on the West End. So those areas were definitely more at risk for environmental sources and like environmental hazards nearby. Socioeconomic status also put them more at risk. So you can see a pretty clear divide there between the West and the East End of Louisville.
Wow, thanks for sharing this insight.
Do you mind, Rebecca, the redlining of Louisville is certainly a very interesting topic, but Also, there's, I guess, built into that as well, like the systematic sort of segregation. Railroads and interstates, and there's so many different barriers, and you can see as soon as you cross over to the other side, there's clear differences in the canopy cover of trees or the built environment, like whether or not there is an abandoned building with busted out windows. All it takes is getting to the other side of the tracks in a lot of parts of Louisville to see that. Were you mapping those sorts of aspects of the built environment as well in your GIS system, or is it primarily noise, or it's just like all the variables you can kind of imagine that affect your well-being?
Yeah, so roads and like proximity to like highways and interstates was definitely taken into account because of noise pollution, especially from bigger interstates and things. We also took into account like, built environment wise, I'm trying to think, we took into account like your proximity to power plants and chemical plants, your proximity to like landfills, which also is why Rubbertown was so heavily at risk, because that's where a lot of those polluting facilities are kind of concentrated. Yeah, you can definitely see it because the West End is more, it's a lot denser, especially in terms of like housing and economic and chemical and factory like activity basically. So definitely the density of that and just the urban environment there contributed a lot to the risk factors.
Do you, kind of tangential, but do you factor in like whether roads are one way or two way or speed limits or anything like that? Or is that sort of that's a higher layer that isn't quite yet in there in GIS?
Yeah, we didn't, but that's a really good kind of next step to look into, because that's a good point. That definitely makes a difference. Yeah.
Thanks.
Yeah, and Rebecca will be sharing some of these insights at Research Louisville in the coming month, and really excited to see where this project goes that I think has really captured the interest of a lot of people on our team. Daraj, we'll turn over to you to kind of give a summary of what the summer's held for you.
Yeah, so I think the main project I was working on this summer involved wastewater epidemiology. So we found that one of the biggest problems that policymakers faced when sort of putting in these quarantining policies for COVID-19 was that they often had to work with inaccurate testing reporting numbers. So whether that's people not having equal access to testing centers, or a lot of people not getting tested because they're asymptomatic, a lot of the testing numbers we had to work off during the pandemic were very faulty, and it made it very hard for us to deal with the pandemic in an effective manner when it came to policymaking. So there was a solution to this, as Dr. Home and her team at U of L found that when people have COVID-19, they shed COVID-19 mRNA in their fecal matter. So we can, in theory, trace what percentage of the population or how much COVID-19 is in the sewage system of a general area, which is a very powerful tool because we have the potential to instantly test the entire population essentially and find what proportion of the people living in this area of the testing facility have COVID-19. The issue, though, is that in order for us to convert these COVID-19 mRNA samples from the wastewater, we have to find out how much water actually passed through the sampling site in order to dilute the amount of the COVID-19 mRNA. And this is a very tricky question, as the equipment required to measure flow rate is very expensive and not very feasible to implement at these testing sites. So what I did over the summer was I created a machine learning algorithm that could use variables such as population density, temperature, precipitation, et cetera, to predict the amount of flow rate that would pass through any site. And we were able to successfully turn these variables into an accurate flow rate prediction, which we could then use in that COVID-19 incidence proportion formula.
Yeah, that is a super worthy topic of focus and discussion and research. And I know a kind of a question I think I've been kind of posing to you and some other people on our team over the summer is thinking about like the applications of wastewater epidemiology. We have flu season, we've living through a COVID pandemic. Can you talk a little bit more about like kind of the application of wastewater epidemiology and where we might be able to go with like the types of tools and methods that you just described?
Definitely. So I think one of the biggest issues with wastewater epidemiology in the past was its efficacy, right? Like the predictions weren't very effective. The predictive tools didn't really apply to multiple populations, but we found that this project breaks all of those barriers. The algorithm we developed has a 92% accuracy rate when predicting flow rate, which is very high and is usable when it comes to determining this COVID-19 incidence proportion rate. And while, hopefully COVID-19 won't be a big issue in the future, Lainey, you are right. Like there is a lot of potential for wastewater epidemiology when it comes to predicting flu season, if any other big pandemics come in the future, and also just determining the general health of a population, because we shed a lot of our bacteria, a lot of health indicators in our fecal material. So having these testing facilities, having these wastewater algorithms is a very powerful tool we can use to determine the health of the entire community.
So Raj, I have a very specific follow-up question. The work that you're doing is absolutely fascinating, and to be a high school senior with this under your belt is incredible. So congratulations on this great work. My question to you is, thinking about some of the different variants of COVID, some of them are more or less associated with gastrointestinal symptoms. So are there ways that you're accounting for this in your algorithm that you've created, or are there ways that this is being thought through in terms of our ability to understand COVID spread in the wastewater?
Yeah, that's a really good question. I think so. The algorithm I developed was mainly just for flow rate. So it doesn't actually look at COVID. It's just, how can we determine how much water passed through this manhole from a given time period, which has a lot of uses. Determining COVID rates, is one of them, of course. And, that is a really interesting question. I think when this study was conducted, it was about Delta was the most common variant. So we didn't really run into these issues. But now, looking back, or right now, there are far more variants, right? And all of them have these varying levels of gastrointestinal significance, right? So that is something we keep in mind. And that's something we also have to keep in mind when applying this algorithm to other, pathogens like the flu, like, the common cold, et cetera, et cetera.
Fascinating. Thanks, Raj.
I appreciate everyone taking the time to describe the work that they've been doing. And you've heard about physiology, you've heard about chemistry, you've heard about physics, sociology, geography, and just so many realms of science and thinking about the world covered in these three experiences over the summer, which is just really exciting to think about how we can all work together to build a healthier city, healthier kind of world. And I think this has been touched on in some of the questions that have been posed, like while you were describing your summer. But if you had to give a few sentence summary of the real world application of your research, we've heard a lot about some technical terms, some new chemicals, flow rates, all of these different things, but in very simple terms, like how do you see the work that you've done this summer applying itself to our individual and our collective health? And Rebecca, I don't think we started with you, so we'll start with Rebecca.
Yeah, so pretty much all of the projects that I've worked on have been aimed at identifying kind of the difference in risk across the city for different things. So your risk of noise pollution, which causes mental and physiological stress, and also its correlation to PTSD. The EVI really, I think the best application is, well, first of all, trying to, and Jirash has also been working on this, trying to show a correlation between these different risk factors and life expectancy, because that's just really powerful information to have. We're also working on making this information available to community members so that they can see the areas that they live in, what puts them more at risk because of where they live. And from there, I think it will be a good tool for people to be able to make risk management plans for the city and for like to tackle specific issues in their area. I think that that would be the best kind of application for this kind of work.
Sure, makes a lot of sense. Ibrahim, we will jump to you.
Awesome. So I mentioned earlier that Myosin has two main configurations, SRX, DRX, where DRX is faster than SRX. And so what I did was I basically tested how carnosine would change this kind of ratio between SRX and DRX. And I found that carnosine significantly increased the levels of DRX. So it kind of made hearts pump faster. But not only did it increase DRX, I also tested how it changed how fast DRX was. And carnosine increased basically both how fast the DRX myosin was and the level of DRX myosin. So it's kind of, I don't want to like, it's like very like, like foundational research, but like in the like long future, after we've done more tests, hopefully this could be used for treatment potentially. Like the certain heart conditions have like much higher levels of SRX, which could potentially cause like a heart that's not working as efficiently since it's not in that like optimal SRX DRX ratio. So carnosine could be used to kind of correct that imbalance in the ratio and kind of make the heart work more efficiently. But like I said, that's like in the like long term. Short term, I want to look more at how it affects carnosine affects heart failure, because heart failure is like heart disease in general are like a leading cause of death in the US and around the world. So I really want to focus on how carnosine, this like small dipeptide, can affect the heart and kind of help hearts that are kind of failing or in heart failure, help them recover, is what I'm trying to say.
No, I think that was a beautiful summary. And Duraj, just in case anyone forgot that we're talking to students, I think I had to hop off for class, but I'm sure he has some thoughts too about the application of his work. And yeah, just shows very much how much of this process is about learning and growing. I think in terms of a final question, one that's kind of reflective in nature, we've kind of worked through what the work was, but it's another question entirely to ask like how it's impacted you as a person and your growth and your journey. So really, I would be curious to learn from each of you how this summer has changed your view of research, of science, of future plans, and really like when you take all of this into perspective, how and has it changed your path forward maybe? And Ibrahim, I know we just spoke to you, but maybe we'll start with you.
No problem. So I had done some like research in high school, like at some like labs, but I was never really like as involved because I was still in high school. I was only there for like maybe 6 hours a week. But as an HA summer intern, I was there, for 40 hours a week, and I was there for 10 weeks. So as I was there, I really got to talk to a lot of, like master's students, PhD students, and the DOC is such like a lovely community. Like I really like love learning about like other people's lives and like the passion they had for their projects. And I was just like, it was just so great to see how much everyone like loved what they were doing, and how like if you ask them about their project, they could talk for like days about it. It was like really awesome to see. So I kind of just like, I gained a greater appreciation for like humans and science, and like how I as a person can like kind of grow my love for science, and hopefully in the future like do my own research and contribute to the field, and like really just like a deeper appreciation for like all things science and all things research. Like it was just such like an amazing community.
That is awesome to hear.
Yeah, that was really kind, Ibrahim. Just so you know, we're all working on our elevator pitches. So it might seem like we could talk for days, but we're trying to tighten it up a little bit.
I know it's no problem at all, but like sometimes I'll be like in a rush and be like, oh, like let me tell you about like this result. Like I'm like, that's great, but like my centrifuge is almost done. Like I gotta go get my stuff. But like it's awesome to hear about it. I love it.
Yeah, and like underscoring that passion that is really driving all of this work is like a great point to make in this. Rebecca, we'll turn to you on this one.
Yeah, I came in as a biology major, so I had done a lot of like biology research, like hands-on like lab research and things. So when I switched into sustainability, I didn't really know how that research would function. I took an environmental vulnerability course last year that I really, really loved. And so coming to the Envirome and like I just felt really lucky to be working on the EVI project because it just kind of aligned so much with like what I was passionate about. And that really like shaped my perspective of what research could be, especially like the community aspect of something like the EVI. Like it was just really like, I feel lucky to have worked on something that has such an impact on the community that I live in and like has such big implications for change. So it kind of just solidified that I definitely made the right decision and that like this is the kind of work that I want to be doing like as a career. So yeah, really glad to have done that.
Rebecca, could you mention who taught or facilitated the environmental vulnerability course?
Yeah, Dr. Tim Darst at U of L.
Cool.
He's amazing. The course was really, really cool.
Yeah, we'll have to look into that.
Board the Envirom at the end of the course, which is how I got started here.
Yeah, sounds like a great course.
Yeah, it was.
Appreciate everyone's time and letting us learn from the three of you. It's been a complete pleasure to listen. And yeah, at the risk of sounding obvious, there are so many opportunities for students to plug in at the Envirom that it just takes an email or reaching out. I'm sure Rebecca, Ibrahim, Daraj would each be happy to talk to people if that's of interest. And yeah, excited to see what the next summer holds and thankful for the contributions you all have made with the time that you've been with us. I think we'll transition to Lauren now to talk a little bit more about COVID in our community. But again, thankful for everyone's time on this.
Yeah, thank you. I have class soon, so I have to leave, but it was so great talking to everyone. Goodbye.
Thank you, Ibrahim.
Thanks so much, Ibrahim.
Hey everyone, so we've talked a little bit about wastewater already today, but in breaking news, our COVID-19 levels appear to have leveled off this week. Still seeing moderately high values out there in the wastewater. And it's hard to tell now whether this is part of a slow decline or just a stubborn, steady high state. But if we look at past waves, where we are now has some characteristics of a slow decline, which would be welcome news for us all, especially as kids went off to school again this week. So that is a good trend to be seeing out there. As many folks know, we're also tracing the genomics of SARS in wastewater, COVID-19 in wastewater, which just means that we're looking at all the variants. We're continuing to look for emerging variants as well. But the biggest news this week is that the Delta variant just won't go away. So it's making a reappearance at some sewer sheds up to 12%. It does not look like the Omicron and Delta mutations are found together. So we have no reason to think that they might be mixing up. And then for the first time in this pandemic, we've gone several months without news of a new variant on the horizon. So that is welcome news as well. And so national reporting from SCAN and Verily continue to report that there's no presence of monkeypox here in our largest treatment plant, Morris Forman, that treats wastewater from the majority of the city. We have heard from the health department that there are very small pockets of infection, but nothing at the community level. So if there is good news there, it is that, if you're not part of special populations that are more at risk, then you can probably take a few breaths about monkeypox here in Louisville. And then our internal labs are still working on our methods needed to measure polio and monkeypox locally, but we're still working out the kinks in that. So far we can tell the presence of pox in general, but there are lots of pox viruses. So chickenpox, for example. So, that's not exactly helpful for the moment not to be able to tell what kind of variant. So again, this is why that genomic work is so interesting and so important. Right, and that is the wastewater report for the week. Natasha, I'll hand it off to you to talk about upcoming events.
Thanks so much, Lauren. It is very interesting to hear the use of technology to understand the spread of our diseases in the wastewater. And you and Duraj have really laid out the value of this, the benefit that it brings. And so grateful for your update on what's going on with COVID and emerging threats here in Louisville. So I'll quickly share about an upcoming event on September 18th from 3 to 5 p.m. The Speed Cinema presents a Dark Waters film screening event. And so the film Dark Waters stars Mark Ruffalo, and he is playing Rob Billett, who discovered environmental exposures in a West Virginia town where a DuPont facility was located. So there will be a panel about this film, and there will also be a book signing. Rob Billett, the subject of said movie, has authored a book, Exposure, Poisoned Water, Corporate Greed, and One Lawyer's 20-Year Battle Against DuPont. So he'll be there signing the book. This event is free. And again, it takes place September 18th from 3 to 5 p.m. And please visit www.speedmuseum.org to find out more about this event. But we are so grateful for you taking time to spend this hour with us, our Science Communication Office Hour. We have to offer special thanks to the stellar interns, Rebecca, Duraj, Ibrahim. It's certainly been a joy to work with them this summer, to see the work that they produce and to hear how this will shape portions of their future. So we thank them for their work and for taking time to share. And I also want to extend great thanks to our team, Drs. Hellman and Carl, Lauren Anderson, and Lainey Taylor. Please don't hesitate to reach out to us. Leave a comment on this video here on Facebook. You can also visit envirominstitute.org. You can also email us at envirom@louisville.edu, or you can even follow us on Twitter at U of L Envirom. Thank you.